Welcome to Part 3 of our Intracellular Structures series, prepared for the European Society of Human Reproduction and Embryology Clinical Embryologist certification. In the last session, we built a detailed picture of the plasma membrane as a dynamic phospholipid bilayer, fluid, asymmetric, and beautifully organized. But a lipid bilayer on its own is essentially just a beautifully engineered fence. It separates inside from outside, but it cannot do very much without the proteins embedded within it and associated with it. That is exactly where we are headed today. This session completes the plasma membrane story, moving from structural lipids to the proteins that transform the membrane into a true functional interface, one capable of communicating, transporting, recognizing, anchoring, and catalyzing. And as always, we will follow every molecular concept directly into the clinic, because understanding these structures at the molecular level is precisely what distinguishes an excellent embryologist from a competent one. Let's begin. In Part 2, we established the membrane as a dynamic phospholipid bilayer with carefully regulated lipid composition and asymmetry. We examined how cholesterol modulates fluidity, how phosphatidylserine sits exclusively on the inner leaflet, and how lipid asymmetry underpins signaling and membrane identity. We introduced the concept of specialized membrane domains, lipid rafts, and caveolae, as ordered platforms within the broader fluid membrane. If Part 2 was about the architecture of the wall, Part 3 is about everything installed within and around that wall: the gates, the sensors, the scaffolding, and the signaling apparatus. The central insight we build on today is this: the lipid bilayer alone is a passive barrier. It selectively excludes charged and polar molecules by virtue of its hydrophobic core, but it cannot actively regulate what crosses it, cannot receive a hormonal signal, cannot anchor itself to a cytoskeleton, and cannot catalyze a chemical reaction. All of those functions belong to membrane proteins. And in reproductive biology, membrane proteins are absolutely central. They coordinate follicular signaling, enable sperm capacitation, mediate gamete recognition, drive blastocele formation, and determine whether an embryo survives vitrification. Every clinically meaningful event at the membrane involves a protein. As we step through this session, keep asking yourself, what goes wrong here in the clinic, and why? This session covers substantial ground, so let's orient ourselves before diving in. We begin by classifying membrane proteins into their three structural categories: integral, peripheral, and glycosyl phosphatidylinositol-anchored proteins, and examining the structural features that allow proteins to sit stably within the bilayer. From there, we move to function: the four major classes of membrane proteins: transport, anchoring, receptors, and enzymes, and what each contributes to gamete and embryo biology. We will then examine the membrane skeleton, the submembranous scaffold that gives cells their mechanical resilience, and the glycocalyx, the carbohydrate coat on the cell's outer surface that plays a surprisingly central role in gamete recognition. We will revisit lipid rafts and caveolae as signaling microdomains, and explore the junctional complexes that organize cells into tissues. The second half of the session focuses on transport: simple diffusion, osmosis, facilitated diffusion through channels and carriers, and active transport by pumps, with specific examples drawn from oocytes, sperm, and embryos. We close with a dedicated clinical section connecting cholesterol, glycocalyx biology, membrane aging, and cryopreservation to real decisions in the assisted reproductive technology laboratory. There is a great deal here, and all of it matters. Before we move into the content itself, let's be precise about what you should be able to do by the time we finish. These learning objectives are your benchmarks, not just for the European Society of Human Reproduction and Embryology examination, but for your daily practice in the laboratory and clinic. You should be able to classify membrane proteins by type and describe how each interacts with the bilayer, not just name them, but explain the physical and chemical logic behind how each one associates with the membrane. You should be able to describe the four functional categories of membrane proteins and give clinically relevant examples from reproductive medicine. You should be able to describe glycocalyx composition and explain its specific roles in gamete interactions, including why the zona pellucida is essentially a specialized glycocalyx. You should be able to distinguish passive diffusion, facilitated diffusion, and active transport with concrete examples drawn from gametes and embryos. You should be able to explain what lipid rafts and caveolae do, and why their cholesterol dependence matters in the context of cryopreservation and sperm capacitation. And finally, you should be able to apply all of this to clinical scenarios, to look at a case involving poor fertilization, failed blastocele expansion, or suboptimal cryosurvival, and construct a mechanistic explanation grounded in membrane biology. That is the standard we are working towards. Let's get into the science. Not all membrane proteins interact with the bilayer in the same way, and the manner of that interaction has profound consequences for how the protein functions and how it can be regulated. There are three principal classes, and understanding each one requires thinking about the chemistry of the bilayer itself. The first class is integral membrane proteins, also called transmembrane proteins. These are embedded directly within the hydrophobic core of the lipid bilayer. You cannot remove them without using detergents or organic solvents that dissolve the bilayer itself. They are physically threaded through it. They span the membrane either once, which we call single-pass or bitopic proteins, or multiple times, which we call multi-pass or polytopic proteins. The key to their bilayer insertion is a stretch of hydrophobic amino acids, typically arranged as an alpha helix of roughly 20 to 25 residues, whose side chains interact with the fatty acyl tails of the phospholipids. Flanking these hydrophobic segments are hydrophilic regions that face the aqueous environments on either side of the membrane. The amphipathic character of these proteins perfectly mirrors the amphipathic character of the bilayer itself. Examples you will encounter throughout this course include ion channels, glucose transporters, hormone receptors, and the sodium-potassium pump, all integral proteins. The second class is peripheral membrane proteins. These do not penetrate the hydrophobic core at all. Instead, they attach to the membrane surface through electrostatic interactions with lipid head groups, hydrogen bonding with other proteins, or physical association with the cytoskeleton on the cytoplasmic face. Because they are not embedded in the hydrophobic core, they can be released relatively gently by changing pH, adjusting salt concentration, or adding chelating agents, without disrupting the bilayer. Peripheral proteins often serve as cytoskeletal linkers, signaling adapters, or membrane-associated enzymes. Spectrin and ankyrin, which we will discuss shortly in the context of the membrane skeleton, are classic examples. The third class is glycosyl phosphatidylinositol-anchored proteins, named after the lipid anchor that tethers them to the membrane. These proteins have no transmembrane domain whatsoever. Instead, their carboxyl terminus is covalently attached to a glycosyl phosphatidylinositol lipid, which inserts into the outer leaflet of the membrane. Because they reside exclusively on the outer leaflet and are associated with lipid rafts, they are in a prime position for extracellular signaling, cell-cell adhesion, and enzyme activity at the cell surface. The glycosyl phosphatidylinositol anchor also means they can be cleaved by specific enzymes called phospholipases, allowing rapid release from the membrane surface, an elegant regulatory mechanism. Keep this outer leaflet exclusive location in mind when we discuss lipid rafts, because it is precisely where glycosyl phosphatidylinositol-anchored proteins congregate. Let's go deeper into the structural logic of transmembrane proteins, because this architecture underlies so much of what these proteins do. The fundamental challenge for any protein that spans the membrane is this. The interior of the bilayer is profoundly hydrophobic, yet the protein must also present functional domains to the aqueous environments on both sides. Nature's solution is the transmembrane alpha helix, a segment of protein chain rich in amino acids with nonpolar side chains, folded into a tightly wound helix. Within a helix, the hydrophobic side chains point outward and interact with the surrounding fatty acyl chains, while the peptide backbone, which has some polarity, is shielded in the helical interior. It is an extraordinarily elegant structural solution to a thermodynamic challenge. Single-pass, or bitopic, proteins cross the membrane exactly once via one such helix. These proteins are common among cell surface receptors. Think of a receptor with a large ligand-binding domain projecting into the extracellular space, a single alpha helical transmembrane segment threading through the bilayer, and a cytoplasmic tail that interacts with signaling molecules inside the cell. Growth factor receptors and many adhesion molecules follow this basic blueprint. Single-pass architecture is clean and direct. One signal comes in on the outside. One response goes out on the inside. Multipass or polytopic proteins cross the membrane 2 or more times, and this architecture opens up qualitatively new possibilities. When multiple transmembrane helices are arranged together, they can form a central channel or pore lined with hydrophilic residues, creating a water-filled pathway through the otherwise impermeable bilayer. Ion channels and water channels, called aquaporins, are built exactly this way. Multipass architecture also enables conformational coupling between helices. When a ligand binds on one side, it induces a shape change that ripples through the transmembrane helices and alters the protein state on the other side. This is how gated channels open and close, how transporters change conformation to move solutes, and how G-protein-coupled receptors, which contain 7 transmembrane helices, transduce signals across the membrane. 7 transmembrane helix receptors, of which there are hundreds in the human genome, are among the most pharmacologically important proteins in medicine, and several of them are critically relevant to reproductive physiology, as we will encounter when we discuss follicle-stimulating hormone and luteinizing hormone receptors. With the structural classification established, let's turn to what membrane proteins actually do. We can organize their functions into 4 broad categories, and thinking through each one reveals just how extraordinary the membrane is as a functional platform. Transport proteins, our first category, include both channels and carriers. They provide regulated pathways for ions, nutrients, water, and metabolites to cross the hydrophobic bilayer that would otherwise exclude them. We will devote significant time to these in the transport section, but for now, appreciate that without transport proteins, a cell cannot regulate its ion balance, import glucose for energy, maintain its pH, or control its volume. All of these parameters are existentially important for dividing embryos and maturing gametes. Anchoring and structural proteins, our second category, link the membrane to the cytoskeleton inside the cell and to the extracellular matrix outside it. Spectrin and ankyrin connect the bilayer to the internal actin network. Integrins span the membrane and simultaneously bind cytoskeletal components inside and extracellular matrix proteins outside, making them mechanosensors that transmit physical information bidirectionally. Without these anchoring proteins, cells would lose their shape, polarity, and mechanical integrity. In the oocyte, the cortical actin network linked to the olemma through these proteins is what positions the meiotic spindle and anchors the cortical granules, both of which are absolutely essential for fertilization. Receptor proteins, our third category, detect extracellular signals and transduce them into intracellular responses. The signals these receptors detect include hormones such as follicle-stimulating hormone and luteinizing hormone, growth factors, cytokines, and physical ligands. G-protein-coupled receptors and receptor tyrosine kinases are the 2 dominant families, and both are heavily represented in reproductive biology. The luteinizing hormone surge that triggers oocyte maturation acts through a G-protein-coupled receptor on granulosa cells, a 7-transmembrane receptor initiating the entire maturation cascade. Enzyme proteins, our fourth category, catalyze reactions directly at the membrane surface. Of particular note in reproductive biology are the enzymes involved in phosphoinositide metabolism, specifically phospholipase C, which cleaves phosphatidylinositol 4,5 bisphosphate to generate inositol trisphosphate and diacylglycerol, the 2 second messengers central to oocyte activation at fertilization. The sperm-borne enzyme phospholipase C-zeta triggers exactly this pathway in the oocyte, initiating the calcium oscillations that complete meiosis and begin embryonic development. We will return to this pathway in detail shortly. Imagine trying to build a functioning city with streets, utilities, and buildings on a substrate that has no foundations, that flexes and deforms unpredictably with every footstep. The plasma membrane faces a similar engineering challenge. A lipid bilayer alone, while beautiful in its physics, is mechanically fragile. What gives cells their resilience under physical stress? Their ability to resist deformation during transit through tissues, under fluid shear, or during the mechanical manipulations of assisted reproductive technology procedures, is the membrane skeleton. The membrane skeleton is a submembranous protein scaffold on the cytoplasmic face of the plasma membrane. Its core components are spectrin, actin, and chitin, and a set of associated proteins. In erythrocytes, which are the most thoroughly studied model, spectrin forms a two-dimensional hexagonal lattice linked by short actin filaments at junction points, with chitin connecting the whole network to the integral membrane protein band 3. The result is a remarkably elastic meshwork that allows the cell to deform dramatically and spring back without rupture. Other cell types, including neurons, oocytes, and blastomeres, have their own versions of this architecture. The membrane skeleton does two critical things. First, it provides mechanical strength, resisting deformation and shear stress while maintaining cell shape. Second, it organizes functional domains by restricting the lateral diffusion of membrane proteins. Rather than allowing channels, receptors, and adhesion molecules to drift freely across the entire membrane surface, the skeleton creates corrals, defined regions within which proteins cluster and concentrate. This spatial organization is essential for efficient signal transduction and cell polarity. In the oocyte, the cortical actin network linked to the olemma is a critical player at fertilization. Cortical granules, the vesicles whose exocytosis creates the block to polyspermy, are anchored just beneath the olemma by this actin spectrin scaffold. The meiotic spindle, which must complete the second meiotic division after fertilization, is also positioned near the cortex through interactions with this network. When the membrane skeleton is disrupted by chemical insults, temperature extremes, or suboptimal culture conditions, the consequences are not subtle. Cortical granule release becomes disordered, spindle positioning is compromised, and polar body extrusion may fail. These are not abstract molecular events. They manifest as fertilization failure and aneuploidy in the clinical laboratory. Structural integrity at the membrane is not optional. It is a prerequisite for developmental competence. Step outside the lipid bilayer, and you encounter something remarkable: a dense, sugar-rich layer coating the external surface of virtually every animal cell. This is the glycocalyx, and in reproductive biology, it is far more than a passive coating. It is an active participant in some of the most important recognition events in human development. The glycocalyx is composed of three types of carbohydrate-bearing molecules. Glycoproteins are proteins with oligosaccharide chains covalently attached to specific amino acid residues. These chains are relatively short and branched. Proteoglycans are proteins bearing long glycosaminoglycan chains, repeating disaccharide units that can extend considerable distances from the cell surface and carry a high negative charge. And glycolipids are lipids with carbohydrate head groups extending into the extracellular space. Together, these three components form a brush-like layer that can extend tens to hundreds of nanometers beyond the lipid bilayer itself. What does the glycocalyx do? It performs several functions simultaneously. It protects the membrane from mechanical and chemical damage, acting as a physical buffer between the cell surface and its environment. It retains water around the cell through the hydrophilic nature of its carbohydrate chains, lubricating cell surfaces and reducing friction. It prevents nonspecific aggregation between cells, keeping cells apart unless specific recognition events occur. And critically, it provides specific carbohydrate ligands that mediate selective cell-cell recognition, including the interactions between sperm and egg. Now here is where this becomes directly relevant to your clinical practice. The zona pellucida, the thick glycoprotein matrix surrounding the oocyte and early embryo, is essentially a highly specialized, massively elaborated form of glycocalyx. Zona pellucida in humans is composed of 4 glycoproteins designated zona pellucida 1, 2, 3, and 4. The zona pellucida 3 glycoprotein in particular is a primary sperm-binding ligand, and it does so through species-specific carbohydrate recognition. Sperm receptors on the acrosomal surface recognize specific carbohydrate configurations on zona pellucida 3, initiating binding. This is not a nonspecific adhesion. It is a molecularly precise handshake requiring both partners to display the correct carbohydrate codes. After fertilization, cortical granule exocytosis releases enzymes that modify these zona pellucida glycoproteins, hardening the zona and preventing additional sperm from binding. The zona reaction, our primary block to polyspermy. Understanding the glycocalyx is, at its core, understanding how the oocyte controls access to itself. We introduced lipid rafts in Part 2, but now we have the protein vocabulary to understand what these microdomains actually do and why they are so consequential in reproductive cell biology. The central concept is that the membrane is not a uniform fluid. It contains regions of different physical order, and those regions differ dramatically in their protein composition and functional capabilities. Lipid rafts are small, dynamic microdomains enriched in sphingomyelin, gangliosides, and cholesterol. Because these lipids pack more tightly than the surrounding phosphatidylcholine-rich membrane, they form a more ordered phase, sometimes called the liquid-ordered phase, sitting within the broader liquid-disordered bilayer. Think of lipid rafts as islands of relative rigidity in a fluid sea, and on those islands, a specific set of proteins preferentially concentrates. Which proteins? Glycosyl-phosphatidylinositol-anchored proteins, which are exclusively in the outer leaflet, are almost always raft-associated. And this makes sense because glycosyl-phosphatidylinositol anchors closely resemble the lipids that form rafts. On the inner leaflet, SRC family kinases, G proteins, and selected second messenger enzymes concentrate within raft domains. The result is that lipid rafts function as pre-assembled signaling platforms. Rather than relying on the random collision of signaling molecules diffusing across the membrane, cells concentrate the relevant partners within a restricted domain, like putting all the members of a meeting in the same room rather than hoping they bump into each other in a large building. Rafts also participate in endocytosis and vesicular trafficking, sorting raft-associated components into specific vesicles for internalization or secretion. They interact with the cytoskeleton and the membrane skeleton, stabilizing their position at functionally important sites such as cell-cell junctions. Cholesterol depletion, which disrupts raft architecture by removing the structural lipid that defines the liquid-ordered phase, scatters raft-associated proteins across the membrane, and impairs the signaling cascades they support. In sperm, lipid rafts undergo a spectacular reorganization during capacitation. As sperm transit the female reproductive tract and cholesterol is progressively removed from the sperm head membrane, raft-associated proteins redistribute toward the acrosomal region. This spatial reorganization of raft proteins is a prerequisite for the acrosome reaction. Without it, the sperm cannot release its lytic enzymes and cannot penetrate the zona pellucida. The connection between cholesterol efflux, lipid raft remodeling, and fertilization competence is not just biologically elegant. It is clinically important, as we will explore when we discuss male factor infertility and in vitro capacitation protocols. Closely related to lipid rafts, but morphologically distinct, caveolae are specialized membrane domains that have been recognized since the 1950s and continue to reveal new functions. The name means little caves in Latin, and the shape is apt. These are small flask-shaped invaginations of the plasma membrane, typically 50 to 100 nanometers in diameter, with the narrow neck opening to the extracellular space and the rounded base projecting into the cytoplasm. Like lipid rafts, caveolae are enriched in cholesterol and sphingolipids. What physically defines them and distinguishes them from flat lipid rafts is the presence of specific structural proteins, caveolins and caveins, that form a curved cytoplasmic coat and impose the characteristic flask shape on the membrane. Caveolin. One is the principal structural caveolin in non-muscle cells. Its integrity is absolutely dependent on membrane cholesterol. Remove cholesterol with methyl-beta-cyclodextrin, and caveolae flatten and disappear within minutes. This cholesterol dependence has important implications for cryopreservation, where membrane cholesterol can be altered by phase transitions during cooling. What do caveolae do? They serve several distinct functions. In endocytosis, they provide a clathrin-independent pathway for internalizing certain ligands, lipid-modified signaling molecules, and some pathogens. In signaling, they concentrate receptors, G proteins, and kinases in a spatially restricted compartment, allowing exquisitely controlled signal modulation, sometimes amplifying signals by bringing components together, sometimes attenuating signals by sequestering components away from the main membrane. In lipid regulation, they participate in cholesterol trafficking and sensing, essentially acting as cholesterol sensors and storage compartments at the cell surface. And in a function that is often overlooked but mechanically important, caveolae act as membrane reservoirs. Under physical stretch, they flatten and contribute their membrane area to the main bilayer, buffering the cell against rupture. This mechanical buffering role may be particularly relevant in contexts where cells experience significant physical force, which in reproductive medicine includes oocyte denudation, microinjection, and embryo transfer. In reproductive biology, caveolae have been identified in follicular granulosa cells and in developing gametes, where they appear to serve as signaling concentration sites and regulators of lipid homeostasis. Their cholesterol dependence links them directly to the clinical considerations we will discuss regarding membrane cholesterol in assisted reproductive technology. In reproductive biology, caveolae have been identified in follicular granulosa cells and in developing gametes, where they appear to serve as signaling concentration sites and regulators of lipid homeostasis. Their cholesterol dependence links them directly to the clinical considerations we will discuss regarding membrane cholesterol in assisted reproductive technology. Adherens junctions are perhaps the most developmentally significant in early embryology. They are mediated by cadherins, a family of calcium-dependent cell-cell adhesion proteins that engage in homophilic interactions. A cadherin on one cell surface binds to the same type of cadherin on the adjacent cell. The cytoplasmic tail of cadherin is connected through a family of proteins called catenins to the actin cytoskeleton. This cadherin-catenin-actin linkage means that adherens junctions mechanically couple the cytoskeletons of adjacent cells, allowing force to be transmitted across tissue boundaries. In the embryo, E-cadherin drives compaction at the Morlus stage, the process by which blastomeres flatten against one another, maximize contact area, and begin to behave as a coherent tissue rather than a cluster of independent cells. Without functional E-cadherin and adherens junctions, compaction fails, and blastocyst formation cannot proceed. Desmosomes use desmosomal cadherents, specifically desmoglines and desmocolins, to link intermediate filaments rather than actin filaments between adjacent cells. This alternative cytoskeletal connection provides exceptional mechanical strength, which is why desmosomes are particularly abundant in tissues that experience high physical stress, such as skin and cardiac muscle. In the embryo, desmosomes appear later than adherens junctions and contribute to the mechanical integrity of the trophectoderm. Tight junctions, composed of claudins and occludin anchored to a scaffold of proteins, including zonula occludens proteins, do something anatomically unique. They form a belt-like seal around the apex of epithelial cells that physically closes the paracellular space. Water, ions, and solutes cannot pass between cells through tight junctions. They must cross the cell itself, giving the epithelium its selectively permeable character. In the trophectoderm of the blastocyst, tight junctions are absolutely essential. They seal the space between trophectoderm cells, creating a closed cavity, the blastocele, that can be filled by active fluid transport. Without tight junctions, any fluid pumped into the blastocele would simply leak back out through the paracellular space, and blastocyst expansion could not occur. The formation of tight junctions in the trophectoderm is therefore a key developmental milestone. Focal adhesions complete the picture by connecting the cell to the extracellular matrix rather than to neighboring cells. Integrins span the membrane, binding extracellular matrix proteins such as fibronectin and laminin on the outside, while connecting to actin stress fibers through talin, vinculin, paxilin, and focal adhesion kinase on the inside. Focal adhesions allow cells to sense the mechanical properties of their environment, to migrate along matrix substrates, and to generate traction forces. In the embryo, integrin-mediated adhesion to the endometrial matrix is a critical step in implantation. With the junction biology in place, let's now turn to what is arguably the most fundamental function of the plasma membrane, regulating what moves across it. Everything we have discussed about membrane structure, the lipid bilayer, the proteins, the junctions, the glycocalyx, ultimately serves the membrane's defining function, selective permeability. The cell maintains an internal environment radically different from the outside world. Different ion concentrations, different pH, different metabolite levels. It maintains these differences not by being impermeable to everything, but by being selectively permeable, allowing some substances through while excluding others, and doing so under precise regulatory control. The driving forces for membrane transport fall into three categories. A concentration gradient drives net movement of uncharged molecules from high to low concentration. An electrochemical gradient, which combines concentration with electrical charge, drives net movement of ions. And for transport against gradients, an energy input is required, either in the form of adenosine triphosphate hydrolysis or an ionic gradient previously established by a pump. The overview table on this slide organizes all transport mechanisms by these parameters. Simple diffusion requires no protein and no energy. Molecules move down their concentration gradient through the lipid bilayer itself. Osmosis is a special case of simple diffusion involving water movement across a selectively permeable membrane. Facilitated diffusion uses specific membrane proteins, channels, or carriers to move solutes down their gradients without energy input. Active transport requires either adenosine triphosphate directly, primary active transport, or the energy stored in an ionic gradient, secondary active transport, to move solutes against their gradients. And vesicular transport, which we will address in later sessions, uses adenosine triphosphate-dependent coat proteins to enclose cargo in membrane-bound vesicles for bulk movement across or between membranes. Notice the logical structure here. The more ordered and specific the transport, moving a particular solute against its gradient, the more molecular machinery is required. Simple diffusion needs nothing but a concentration gradient and a permeable membrane. Active transport of a specific ion against its gradient requires a dedicated pump protein, a source of energy, and often precise regulatory control of pump activity. The cell invests more molecular complexity wherever tighter control is needed. As we examine each mechanism in turn, we will see exactly why each type of transport appears where it does in gamete and embryo biology. Simple diffusion is the thermodynamic baseline against which all other transport mechanisms are defined. A molecule in solution moves randomly due to thermal energy, and when there is a concentration gradient, the statistical result of that random movement is net flux from high to low concentration. No protein is needed, no energy is consumed. The gradient itself is the driving force, and it runs down until equilibrium is reached. Which molecules can cross the bilayer by simple diffusion? The rules are governed by the hydrophobic core of the membrane. Small, nonpolar molecules dissolve readily in the lipid interior and cross quickly. Oxygen, carbon dioxide, nitrogen, and steroid hormones all cross by simple diffusion. This is clinically important. Steroid hormones, including estradiol, progesterone, and testosterone, enter their target cells by diffusing directly through the membrane, not through protein transporters. Small, uncharged polar molecules can also cross by simple diffusion, though more slowly. Water, ethanol, and glycerol fall into this category. However, larger polar molecules such as glucose and amino acids cannot cross by simple diffusion. Their polarity makes them poorly soluble in the hydrophobic bilayer core. And essentially all ions, sodium, potassium, calcium, chloride, hydrogen, are completely excluded by simple diffusion regardless of concentration gradient. The nonpolar bilayer core presents an enormous energetic barrier to charged species. This is why ion channels and pumps are not evolutionary luxuries, but absolute necessities. Osmosis deserves special attention in our clinical context. It is the net movement of water across a semi-permeable membrane driven by a solute concentration gradient, technically, by differences in water activity or water chemical potential. Water moves from where solute concentration is lower, more water activity, to where solute concentration is higher, less water activity. Osmosis can occur by simple diffusion through the bilayer, but this is relatively slow. The much faster and physiologically dominant pathway is through aquaporin channels, dedicated water channel proteins that can facilitate water movement at rates orders of magnitude faster than bilayer diffusion alone. Why does this matter clinically? Because osmotic stress during cryoprotectant loading and unloading is one of the most significant sources of damage in oocyte and embryo vitrification. When a cryoprotectant solution is added to cells, the cells initially lose water rapidly due to the high external osmolarity, shrinking dramatically. Then, as the cryoprotectant slowly permeates the membrane, water re-enters and volume recovers. On warming and dilution, the process reverses. Each of these osmotic excursions subjects the membrane and organelles to mechanical stress, and exceeding safe volume limits causes irreversible damage. Understanding osmotic kinetics, aquaporin expression, and cryoprotectant permeability is not an academic exercise. It is the mechanistic foundation for designing safe and effective vitrification and warming protocols. Beyond simple diffusion lies the world of protein-mediated transport, where specific membrane proteins provide regulated pathways for molecules that cannot cross the bilayer on their own. This is where the cell gains genuine control over its internal composition. Facilitated diffusion uses channel proteins or carrier proteins to move solutes down their concentration or electrochemical gradients. No energy expenditure required, but specific proteins are absolutely necessary. Channel proteins form water-filled pores through the membrane, allowing rapid movement of specific ions or water molecules. These channels are gated. They open and close in response to specific signals such as membrane voltage, ligand binding, or mechanical force. Voltage-gated calcium channels, potassium channels, and aquaporins are all examples of channel proteins. Carrier or transporter proteins work differently. Rather than forming an open pore, they bind their specific solute, undergo a conformational change, and release the solute on the other side. This binding and releasing mechanism makes carriers intrinsically slower than channels, but allows more precise specificity and tighter regulatory control. Carriers are classified by the number and direction of solutes they move. A uniporter moves one type of solute in one direction. The glucose transporter family, called glucose transporter proteins, are uniporters that move glucose from high to low concentration into cells. A symporter moves two solutes in the same direction simultaneously. The sodium-glucose cotransporter uses the inward sodium gradient to drive glucose uptake against its own concentration gradient. An antiporter moves two solutes in opposite directions. The sodium-calcium exchanger extrudes calcium from the cell while sodium enters. Active transport, the ability to move solutes against their electrochemical gradients, requires energy input. Primary active transport couples transport directly to adenosine triphosphate hydrolysis. The sodium-potassium pump, or sodium-potassium adenosine triphosphatase, is the paradigmatic example. It uses one molecule of adenosine triphosphate to export 3 sodium ions and import 2 potassium ions, both against their respective gradients, establishing the ion asymmetries that underpin cell volume regulation, membrane potential, and the driving force for secondary active transport. Calcium adenosine triphosphatase, including the sarcoendoplasmic reticulum calcium adenosine triphosphatase, known as the SERCA pump, actively transports calcium back into the endoplasmic reticulum against its concentration gradient, replenishing calcium stores between calcium release events. Secondary active transport, by contrast, uses the gradient established by a primary pump as the energy source for a second solute. No direct adenosine triphosphate coupling, but the energy was provided upstream by the pump that built the gradient. The sodium-potassium adenosine triphosphatase in the trophectoderm of the developing blastocyst deserves special mention. This pump is responsible for blastocele formation. By actively pumping sodium into the blastocele cavity, it creates an osmotic gradient that draws water in through aquaporins, expanding the cavity progressively. Inhibit this pump, and blastocele formation stops. The pump's activity is therefore a marker of trophectoderm differentiation and a prerequisite for blastocyst development, a fact we return to when we discuss embryo selection. Let's now bring these transport mechanisms to life in the context of your daily laboratory work, where each of these molecular events has a direct clinical correlate. Start with calcium signaling in the oocyte. At fertilization, the sperm delivers phospholipase C-zeta into the oocyte cytoplasm. This enzyme cleaves phosphatidylinositol 4,5-bisphosphate in the plasma membrane to generate inositol trisphosphate, which binds to inositol trisphosphate-gated calcium channels on the endoplasmic reticulum membrane and triggers calcium release into the cytoplasm. This calcium release propagates as a wave across the oocyte. Then the sarcoendoplasmic reticulum calcium adenosine triphosphatase pump actively reloads the endoplasmic reticulum calcium stores, using adenosine triphosphate generated by mitochondria, and a second wave is triggered. This oscillatory pattern of calcium release and reuptake continues for several hours after fertilization and is absolutely essential for oocyte activation, cortical granule exocytosis, and completion of the second meiotic division. If the mitochondria are dysfunctional and cannot supply adequate adenosine triphosphate, the sarcoendoplasmic reticulum calcium adenosine triphosphatase pump fails to reload the stores, calcium oscillations are blunted, and oocyte activation is incomplete. This is one reason why mitochondrial health is a predictor of oocyte developmental competence. In the blastocyst, the sodium-potassium adenosine triphosphatase in the trophectoderm drives blastocele expansion through the mechanism we just described. Aquaporins, specifically aquaporin 3 and aquaporin 7 in oocytes and early embryos, facilitate the rapid water movement necessary for osmotic equilibration during cryoprotectant loading. Glucose transport shifts progressively during pre-implantation development. Early cleavage embryos rely primarily on pyruvate and lactate as carbon sources, with glucose uptake being low. Around compaction, glucose transporter expression increases and glucose utilization rises substantially. This metabolic transition is reflected in the changing nutrient requirements of sequential embryo culture media, which typically provide higher glucose concentrations in the blastocyst medium than in the cleavage stage medium. These are not abstract biochemical facts. They explain why your culture media formulations matter, why mitochondrial function is a determinant of developmental potential, and why aquaporin biology is directly relevant to your cryopreservation outcomes. Every transport event we have described has a clinical handle, a protocol adjustment, a quality indicator, or a diagnostic tool that connects the molecular mechanism to what you observe in the laboratory. Cholesterol sits at the intersection of membrane structure, cell signaling, and reproductive competence in ways that are only now being fully appreciated. For the clinical embryologist, cholesterol biology is not a background detail. It is a practical consideration in understanding gamete quality, capacitation, and cryopreservation outcomes. Begin with the oocyte. The olemma requires precise cholesterol content to maintain its structural and functional integrity. Cholesterol stabilizes the membrane, maintains the organization of lipid rafts, and provides the correct physical environment for the clustering of sperm receptors, the proteins on the olemma that mediate sperm-egg binding and fusion. Here is a striking fact. Oocytes are cholesterol auxotrophs. This elegant term from microbiology means they cannot synthesize sufficient cholesterol for their own needs. The oocyte is metabolically dependent on the surrounding cumulus cells and on cholesterol delivered from follicular fluid, predominantly through high-density lipoprotein-mediated transport. If high-density lipoprotein function is disrupted, or if cholesterol transport pathways are impaired, as they may be in metabolic syndrome or polycystic ovary syndrome, the oocyte membrane does not receive adequate cholesterol. The consequences include impaired meiotic control, disordered oocyte activation, and compromised embryo competence. Treating membrane cholesterol adequacy as a clinical variable opens new perspectives on why oocytes from some patient populations underperform despite apparently normal morphology. During cryopreservation, membrane cholesterol modulates resistance to the phase transitions that occur as temperature drops. As cells are cooled, the bilayer can transition from a liquid crystalline to a gel phase, a shift that can cause lipid phase separation and membrane disruption, particularly at the boundaries between cholesterol-rich and cholesterol-poor domains. Cholesterol, by its effect on membrane fluidity, broadens the temperature range over which the membrane remains in an appropriate physical state and resists these damaging transitions. This is why modulating membrane cholesterol in cryopreservation media, by supplementing with cholesterol-loaded cyclodextrin carriers, can improve oocyte, sperm, and embryo survival rates by preserving raft architecture through the vitrification and warming process. Sperm capacitation provides perhaps the most beautifully worked-out example of cholesterol's role in reproductive biology. When sperm are deposited in the female reproductive tract, they are not yet fully competent to fertilize. They must undergo capacitation, a complex remodeling program that prepares them for the acrosome reaction. A critical early event in capacitation is the selective efflux of cholesterol from the sperm head membrane into cholesterol acceptors in the female reproductive fluid, primarily albumin and high-density lipoprotein particles. This cholesterol removal increases the fluidity of the sperm head bilayer and triggers a lateral redistribution of proteins, including raft-associated signaling molecules, toward the acrosomal region. The resulting changes in membrane organization prime the acrosome reaction machinery. When the sperm subsequently encounters zona pellucida 3 protein at the egg surface, the acrosome can fuse with the plasma membrane and release its lytic enzymes. Defective cholesterol efflux means defective capacitation, which means the acrosome reaction cannot occur, and fertilization fails despite apparently normal sperm morphology and motility. In vitro capacitation protocols for intrauterine insemination and in vitro fertilization use albumin or methyl beta-cyclodextrin to mimic this physiological cholesterol removal, a direct clinical application of this molecular mechanism. When you add albumin to your sperm preparation medium, you are facilitating cholesterol efflux, remodeling lipid rafts, and preparing the acrosomal machinery for fertilization. The plasma membrane's glycocalyx and lipid composition do not merely influence fertilization. They are active variables in embryo quality assessment and in determining clinical outcomes across the entire spectrum of patient ages and conditions. The olemma and early embryo surface carry a rich glycocalyx decorated with integrins, mucins, gangliosides, glycoproteins, and glycolipids. At fertilization, this glycocalyx mediates the species-specific recognition between sperm and egg. The zona pellucida glycoproteins 1 through 4 are the primary mediators, with zona pellucida 3 and 4 being the principal sperm-binding ligands. Their specific carbohydrate configurations act as molecular identity tags, ensuring that only conspecific sperm can initiate binding and trigger the acrosome reaction. Beyond species specificity, the zona pellucida also functions as a physical and immune barrier during pre-implantation development, modulating maternal immune tolerance to the embryo, a poorly understood but clinically relevant phenomenon in recurrent implantation failure. The relationship between membrane fluidity and reproductive aging is one of the most clinically important stories in modern reproductive medicine. With advancing maternal age, reactive oxygen species accumulate in the follicular environment and inflict damage on the polyunsaturated fatty acids that are abundant in oocyte membranes. Lipid peroxidation converts flexible, fluid membranes into stiffer, more viscous structures. The consequences cascade through every membrane-dependent event in oocyte and embryo biology. Sperm-oocyte fusion is less efficient because membrane merger requires adequate fluidity. Cortical granule exocytosis is impaired because vesicle fusion with the lemma is slowed. Cytokinesis, the physical division of the cell, requires membrane deformation that stiff membranes resist. Compaction depends on E-cadherin-mediated adhesion that is modulated by membrane fluidity and blastocele formation. Via the sodium-potassium adenosine triphosphatase depends on trophectoderm membranes with appropriate physical properties. The cumulative result is lower fertilization rates, higher aneuploidy, and poorer blastocyst development in older patients, outcomes that are at least partly attributable to membrane-level changes. This is why antioxidant supplementation strategies, melatonin, coenzyme Q10, and vitamin E in culture media and as oral supplements, and the use of 5% oxygen culture conditions rather than atmospheric 21% oxygen, are not merely experimental niceties. They are attempts to limit reactive oxygen species-mediated membrane damage in a patient population that is intrinsically more vulnerable to oxidative stress. Finally, the externalization of phosphatidylserine, the phospholipid normally confined to the inner leaflet, is a well-established signal of apoptosis. When cells undergo programmed cell death, phosphatidylserine flips to the outer leaflet and serves as an eat me signal for phagocytes. In embryos, phosphatidylserine externalization reflects compromised cellular viability. Researchers are currently investigating whether phosphatidylserine exposure on the embryo surface can be detected non-invasively, without penetrating the zona pellucida, as a marker for embryo selection. If validated, this would give us a membrane-level viability readout to complement morphological grading and morphokinetics. The membrane, it turns out, may literally signal its own health status if we know how to read it. Our final clinical slide brings together transport proteins, structural integrity, and membrane function as quality parameters, the kind of parameters that inform both our laboratory decisions and our assessment of gamete and embryo viability. Consider the interconnectedness of membrane transport and embryo development. Transporters and receptors control the ionic milieu inside cells, the availability of nutrients, and the responsiveness to signals. But the membrane is not just a biochemical interface, it is also a mechanical structure. The connections between transport proteins, the actin cytoskeleton, and spectrin form an integrated mechanical system. When this system is disrupted, whether by oxidative damage, osmotic injury, or inappropriate culture conditions, the consequences extend beyond just transport. The shape of the oocyte and blastomere, the positioning of organelles, and the mechanical resilience during manipulation all depend on membrane structural integrity. A membrane that has lost its structural coherence will show downstream failure in maturation, fertilization, cleavage, compaction, and potentially blastocyst expansion. Viewing membrane integrity as a multidimensional quality parameter, not just a structural feature, but a functional prerequisite for every downstream developmental event, is the right conceptual frame. Sperm mitochondrial membrane potential provides one of the most direct functional readouts of sperm quality currently available. The mitochondrial membrane potential, which reflects the electrochemical gradient across the inner mitochondrial membrane, is a measure of mitochondrial activity and adenosine triphosphate generation capacity. It can be quantified using fluorescent dyes such as JC1, which changes fluorescence emission depending on membrane potential. Sperm with high mitochondrial membrane potential show better motility, higher fertilization capacity, and lower deoxyribonucleic acid fragmentation rates. This makes mitochondrial membrane potential a functional fertility marker applicable to sperm selection for assisted reproductive technology, a practical tool for going beyond standard motility and morphology assessment. Cryopreservation brings all of these membrane considerations together under maximum stress. For a cryoprotectant agent to protect against intracellular ice formation, it must permeate the cell membrane. It must cross the lipid bilayer and enter the cytoplasm before cooling begins. The rate of cryoprotectant permeation depends on temperature, which affects membrane fluidity and therefore permeability, and on aquaporin expression, which facilitates water movement during the osmotic shifts that accompany cryoprotectant loading. Post-warming, membrane integrity can be assessed by dye exclusion tests, which measure whether the membrane has maintained its selective permeability, as well as by fertilization rates and morphokinetic normality in subsequent embryo development. The entire science of cryoprotocol optimization, choosing the right cryoprotectant type and concentration, setting the right exposure time and temperature, designing the dilution steps, is fundamentally a science of membrane biophysics. Every parameter in your vitrification protocol has a membrane rationale. We have covered substantial ground in this session, and before we close, let's consolidate the key concepts that you should carry forward from Part 3. Membrane proteins transform the lipid bilayer from a passive barrier into an active functional interface. The three structural classes, integral proteins embedded in the hydrophobic core, peripheral proteins associated with the surface, and glycosyl phosphatidylinositol-anchored proteins tethered to the outer leaflet, each have distinct interaction modes with the bilayer that determine their function and regulation. The four functional categories, transport, anchoring, receptors, and enzymes, cover every active process at the membrane. In gametes and embryos, transport proteins regulate ion balance, nutrient uptake, and pH. Anchoring proteins maintain cortical architecture and spindle positioning. Receptors transduce follicular signals and sperm activation cues. And enzymes including phospholipase C-zeta generate the calcium signals that activate the oocyte at fertilization. The glycocalyx and zona pellucida are specialized extracellular carbohydrate structures that mediate gamete recognition, establish species specificity of fertilization, and contribute to the block to polyspermy through modification of zona pellucida glycoproteins after cortical granule exocytosis. Lipid rafts and caveolae organize signaling and trafficking into spatially defined microdomains. Their cholesterol dependence is a key vulnerability. Cholesterol depletion disperses raft-associated signaling platforms and flattens caveolae, with direct consequences for capacitation, acrosome reaction readiness, and cryopreservation survival. Transport across membranes ranges from simple diffusion of nonpolar molecules to adenosine triphosphate-driven active transport against gradients. The sodium-potassium adenosine triphosphatase in the trifectoderm is essential for blastocele formation. Calcium channels and sarcoendoplasmic reticulum calcium adenosine triphosphatase pumps generate and sustain the calcium oscillations that activate the oocyte, and aquaporins determine water flux rates during osmotic challenges in vitrification. Cholesterol efflux during sperm capacitation is mechanistically required for acrosome reaction competence. In vitro capacitation protocols that use albumin or methyl beta-cyclodextrin are directly mimicking this cholesterol removal step. And finally, membrane integrity, fluidity, and lipid composition are modifiable clinical parameters. Antioxidant supplementation, 5% oxygen culture, optimized cryoprotocol design, and awareness of patient metabolic factors that impair cholesterol transport are all interventions grounded in membrane biology. Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.